And welcome everyone. We're delighted to have you join us for today's webinar from Tevard Biosciences, a new name to some of you on today's call. Tevard are developing unique tRNA-based therapies to target nonsense mutations. This is an important approach to treat the approximately 12 to 14% of Duchenne patients who harbor a premature stop codon in the dystrophin gene and have limited treatment options. For full disclosure, Cure Duchenne first invested in Tevard in 2023, and again this year to help accelerate and advance this work. I'm Michael Kelly. I'm the Chief Scientific Officer at Cure Duchenne, and I'm going to get us started with some housekeeping. First, you have an opportunity to ask questions today. You can click on the Q&A button at the bottom of your screen, submit a question anytime throughout the presentation. We'll have a moderated Q&A session at the end of the webinar, and I'll be joined by my colleague, Dr. Liana Orlando, Cure Duchenne's Vice President of Research. For those questions that we're not able to address, we'll work with the Tevard team and follow up. And finally, after today, this webinar will be available on Cure Duchenne's website as a community resource. So let me begin. The team at Tevard are developing a unique approach to target nonsense mutations based on modified suppressor tRNAs that specifically target premature stop codons. The resulting read-through is designed to replace the missing amino acid and produce full-length natural dystrophin. To learn more about this, we have Daniel Fisher, Tevard's CEO, and his team with us today to introduce the technology and present some of their latest results. So without further ado, let me hand it over to Daniel to get us started.
Thank you, Michael, and thank you for everybody that's here listening to our story and our very exciting technology. So with that, let me start with okay. First of all, as background, we're a company started by renowned scientists and parents who have children with rare diseases. I'm one of the founders of the company. I have a daughter with a disease called Dravet syndrome, which is a very rare and severe type of genetic epilepsy. If you're wondering where does a strange name Tevard come from, it's basically reversing Dravet. Our original goal was to reverse Dravet syndrome, was to create a type of gene therapy that would allow us to target the root cause of the disease. And when we developed the technology, we realized actually that we had a broad platform that could work for many other diseases caused by mutations, especially caused by large genes where you can't apply traditional gene therapy because you can't deliver this gene using traditional methods. So our goal is to get our therapeutic to as many patients, to as many indications as possible. And since we're founded and ran by parents with children with rare diseases, we understand the urgency and the need. So trust us, we're doing our best to get the therapy to the clinic as quick as possible. But of course, we want not only a very efficacious therapy, but also a very safe therapy. We're also extremely grateful to Cure Duchenne for being our partners in this endeavor since we started thinking about working on DMD. Just to mention also that picture, it's an old picture of my twin girls. The one on the left in purple is my daughter, Natasha, who has Dravet. So with that, what we're doing at Tevard, we're looking at working on an approach that will revolutionize gene therapy. This approach will allow us to address 35 million patients worldwide suffering of a rare disease caused by nonsense mutations. Across multiple indications, different number of patients are affected by a nonsense mutation. As Michael mentioned, in Duchenne muscular dystrophy, around probably 12 to 15% of all cases are caused by nonsense mutations. And nonsense mutations can be divided in three types: a TGA mutation, a TAG mutation, and a TAA mutation. The beauty and the exciting part of this technology is it actually allows us to produce a full-length functional dystrophin as opposed to a shorter version of the protein. Next. Some of us are here, but we have a very experienced management team that has experience actually getting other gene therapies to the clinic. So again, we have the expertise in place to get to the clinic as soon as possible. And we also have a pretty amazing group of scientific advisors and board members that also not only assist us in the day-to-day in achieving our mission, but they're extremely excited by the technology, by the data that they're currently seeing. And let me start with some basic biology, even if I'm not a biologist, so I'll try and do my best here. So you have what we call the central dogma in biology, where you have initially DNA, which is the instruction set. By transcription, that DNA transmits the same message to the mRNA. And through translation, the mRNA produces protein. So in the specific case of dystrophin, in the DNA, you have the dystrophin gene, and the protein, of course, is a dystrophin protein, which is missing in Duchenne patients. Now, if we focus on the middle part, which is translation in the mRNA, you have three key components. You have the mRNA, which is a message. Then you have the ribosome, which is that big piece that you see in the middle, which goes across the mRNA and reads those three-letter codes or codons. And in order to read the three-letter code, it finds a tRNA or a transfer tRNA, which are those little pieces floating around, which have a small ball, which is an amino acid. So basically what the ribosome does, it finds a tRNA that matches that three-letter code, binds to it, places that little amino acid in that growing chain, which is called a polypeptide chain. And when the ribosome gets to the end of the message, you have a full-length protein that has been created, and it's a functional protein. And in that code, the three-letter codes, you have 61 codes, which represent one specific amino acid, and you have three different codons or codes, which are a stop codon. And those stop codons, naturally in a normal message, are at the end of the message. And when the ribosome gets to the end, it basically tells the ribosome, stop here, the message stops here, release the protein. Now, the problem arises when you have a single point mutation in what's supposed to be a sense codon, and that sense codon becomes a premature stop codon. The ribosome gets to the premature stop codon. It cannot find a tRNA that matches that. The system falls apart. It's called nonsense-mediated decay. And where before you should have a full-length protein, you have a truncated, non-functional protein, which basically, of course, doesn't work. So what we do, in essence, it sounds pretty simple. We take a tRNA, we modify the anticodon, the bottom part of the tRNA, so it can recognize a premature stop codon. So what happens now, you have the ribosome reading over the message, gets to the premature stop codon, finds a modified engineered tRNA, places the amino acid in the polypeptide chain, and produces a full-length protein. So where before you had a mutated DNA producing a mutated mRNA, unable to produce a protein, now you have the same mutated DNA, the same mutated mRNA, but now being able to produce actually a full-length, a complete functional protein, in this case, a complete and functional dystrophin. And with that, I'll introduce my colleague, our CSO, Elizabeth Gardner, which will continue explaining the technology.
Thanks, Daniel. And thanks, everyone, for being on the call today. So suppressor tRNA technology looks pretty simple. So essentially, you have suppressor tRNAs, which actually recognize this nonsense mutation as an actual codon. And this actually offers a way to really abstract from a gene-centric approach to a premature termination codon-centric approach. This allows us to really attack all of the potential nonsense mutations that may exist in a particular disease state. We've been able to develop a few highly efficacious therapeutic suppressor tRNAs, and we believe that this can cover thousands of rare diseases, including Duchenne muscular dystrophy that is caused by nonsense mutation. The advantage of this approach, as Daniel has explained, is that we're using native cellular machinery to actually produce our transgene, in this case, the tRNA, and we're restoring full-length protein using the cell's native machinery. This avoids transgene overexpression that could potentially be detrimental to the heart or to the skeletal muscle. And in this case, our suppressor tRNAs are specific for nonsense mutations. They do not promote native stop codon read-through, and they're not doing anything aberrant in the cell. Essentially, what they do is they see, recognize, deliver the correct amino acid, and allow the production of the full-length protein. We do this with AAV, which has been shown to be therapeutically beneficial in other DMD therapeutics. We deliver specifically our suppressor tRNA gene. This gets produced in a tRNA by the cell, and then it's this tRNA that now interacts with the cell's machinery in order to produce the full-length dystrophin. This has a lot of advantages over current therapies, specifically because it doesn't rely on stuffing a rather large transgene into the AAV. We work with a very small tRNA, and in this case, it allows us to address all the different nonsense mutations that the gene may have. This allows us to really take a single drug and into one development program, and then look across all the potential mutations that exist. As Daniel mentioned, there's 3 different ways of doing this: the TAA, the TGA, and then the TAG. And you may have this mutation, and we would be able to address it with very specific tRNAs in what we call an exact correction strategy. Using this approach, we would be able to address about 70% of all the nonsense mutations in the DMD spectrum. And this, as Daniel mentioned, is about 12 to 15% of the DMD patients. The idea behind this, and we've already started talking to regulators, is that we could potentially get one approach approved by the FDA, and we could get preclinical and then clinical safety and efficacy, and then rapidly expand into other muscular dystrophies that would potentially be impacted by nonsense mutations. I know that this is a very tightly knit community, and that sometimes patients may not have Duchenne, they may have Becker's or dysferlinopathies, etc. And so we want to be able to approach any muscular dystrophies that actually have nonsense mutations. So suppressor tRNAs really address a patient population with a very high unmet need. So some patients could actually respond to a tRNA therapeutic, could respond to a microdystrophin therapeutic. And we believe that, these patients would be well served with either. But in the case of a nonsense mutation, specifically the tRNA therapeutic could address your nonsense mutation and then could focus on the production of a full-length protein as opposed to a smaller truncated form of the protein. Patients that can respond to exon skipping technology and tRNA technology are actually minimally overlapping. So you can actually see here, everything in purple shown on this slide is a nonsense mutation that has been identified as being pathogenic in a patient that has Duchenne muscular dystrophy. The red is specifically the exon 44, 45, 51, or 53, and those patients could respond to exon skipping technologies. However, those patients don't actually have nonsense mutations and so could not respond to tRNA therapeutics. We anticipate that these patient populations, while separate, will have good therapies in the future, with exon skipping technologies being so advantageous currently, as well as suppressor tRNAs coming into the clinic. So we believe that full-length dystrophin is optimal for muscle function, largely because of how complicated dystrophin is as a molecule and what it does. So obviously it's interacting with a lot of other proteins. And in particular, it doesn't just interact on a structural sense. It interacts in a signaling sense too, and responds to the inflammatory processes that are happening inside the cell. What we believe with a full-length dystrophin, as you can see here depicted in this cartoon, is that it will really help preserve critical protein-protein interactions required for optimal muscle function and repair. And we believe that because we can restore the full-length dystrophin protein in muscle cells with nonsense mutations, this will provide a safe and efficacious approach. One critical thing is that what we're trying to do is rescue dystrophin in skeletal muscle as well as cardiac muscle. And this is part of all our preclinical efforts, where we're strongly trying to make sure that we're safe in expression of full-length dystrophin in both the skeletal muscle tissue as well as the cardiac muscle tissue. There's other tissues that are obviously very critical. This includes intercostal muscles. This includes diaphragm. These are different muscles that we are focused on as well. And this is another reason why we're using an AAV approach, and specifically using an approach that is focused on myo-specific capsids. So muscle targeting, muscle specific, and liver detargeting approach that will be efficacious and safe for the patient population. I'd like to hand this over now to Peter Iman, who is our Director of Research, to talk about how Tevard has taken this from a nascent idea to a clinically ready approach. Peter?
Thanks, Elizabeth. So yeah, you can see on this slide, it's sort of a slide that, oh, I'm sorry, let me turn my camera on. There we go. So it's a slide that essentially captures the work that we've done in the DMD space over the last couple of years. But I think Elizabeth and Daniel gave you a very nice rundown of the suppressor tRNA technology. And I would say, on the one hand, it sounds fairly straightforward. You take a tRNA and you make it into one that can recognize a nonsense mutation, premature termination codon, and you get a full-length protein. What we found is that in practice, it's not quite that straightforward. So suppressor tRNAs really need a lot of optimization if you want them to be highly efficient. And there's a number of reasons you want them to be efficient. Obviously, you would like as much recovery of the full-length protein as possible in the cells that are supposed to express it. But I think, as we're all aware, you'd also like to be able to do it at very low doses of AAV to ensure the best possible safety profile. So you can see we've got sort of our schematic of the tRNA here on the left-hand side. We're putting this into myotropic AAVs. And then we're initially screening all of these in mouse models, a standard MDX mouse model of dystrophin that you're probably familiar with. And we started off with what we would call our generation 1 suppressor tRNAs. These are sort of the most, in some ways, they're the most basic suppressor you can imagine. You start with an endogenous tRNA, you make a change to its anticodon so it can recognize a nonsense mutation, and you see how well it does in mice. So that's what we'd call generation 1 suppressor tRNAs. And the little graph on the right-hand side here is actual data, in vivo data from a mouse model. We're delivering IM, so we're getting a fairly high dose. We'll show you some IV results later on, but for screening purposes, this gives us a good tool to assess suppressor tRNA activity and find efficient ones. And you can see that when we started working with generation 1 suppressors, we do get some rescue of full-length dystrophin in mice when we deliver with this, but it's quite low. I think the best generation 1 suppressor that we got, still giving under 10% rescue of full-length dystrophin in this particular model and delivery paradigm. And we really wanted to do better than that, and we thought we could. And so we did a number of screening campaigns where we assess a large number of additional modifications made to the suppressor tRNA itself outside of the anticodon that optimize various properties of the suppressor as it interacts with ribosomes, as it interacts with other protein factors. And so this is what we call initially our generation 2 suppressors. And you can see just by making some changes and doing screening, we got a very substantial bump up in rescue, up to about 20%. And this already was sufficient when we took it by intravenous injection for systemic delivery to have a functionally beneficial result. But we were so encouraged by the results that we were getting with this screening, we actually iterated on that and did a couple of additional rounds of screening, taking what we learned from the first round, applying it to a new screening library, as well as making some changes and optimizations to the delivery vector itself. So that's our generation 2, 3, and 4 suppressors. And you can see that with each generation, we get dramatically better rescue of full-length dystrophin at the same dose using the same route of administration. And so this is what really gets us excited, I think. This tells us that we can rescue full-length dystrophin, and we've got the opportunity to do it with AAV delivery at doses that we think will be quite low and still efficacious. So next slide. So here we're still looking at some IM data, but I just want to show you that we are getting rescue of the full-length protein. If we look by Western blot, that's that blot that you see sort of in the top center of the image, where you can see examples of 100% all the way down to about 1% of normal dystrophin. And you can see that what we're rescuing is full length, runs the same size. And by IM injection, we're getting quite good rescue. And if we go to the next slide. So now we've taken this into a much more clinically meaningful experiment. So we're delivering these suppressors now throughout the mouse using intravenous administration. And we're going to be running them through a number of functional assays to see what's actually going on at the level of muscle function. So we've got different readouts, and if you want to click again, and the video will start playing. So what you're going to see in the video is what's called a wire hang assay. And it's just putting the mice through a few simple basic exercises. In this case, they're hanging on to essentially a high wire, and they're going to drop down into their bedding when they can't hold on any longer. So you can see one group labeled untreated and one group labeled scramble treated. So these are essentially the untreated dystrophin mice, and they cannot hang on to this rod for very long. That's why you can see in the video, both of these have now fallen down into their cage and they're running around. But the wild type mouse is hanging on, and importantly, the suppressor tRNA treated mouse is still up there running back and forth along its wire. It's really sort of matching the wild type mouse in terms of its performance. And this is an assay that's going to require coordinated activity of a lot of different muscle groups. We're showing you one mouse in each of these, but you can see in the graphs to the left, this is an effect that you see when you look at populations of mice, and you're getting back to wild type levels in terms of this assay. With the wire hang assay, that's the bar in purple, matching wild type mice in white. And other assays that we've run include grip strength. So this is, as it sounds, it's basically just measuring how much strength the mouse is able to exert when you put it onto a sort of a scale and measure. And you can see in the gray bars, the untreated mice or the control treated mice have a deficit relative to the wild type mice. And this is essentially fully restored when we deliver our suppressor tRNA systemically. And finally, rotarod is one more functional assay that we like to use. In this case, it's measuring how long the mice can maintain on a rotating cylinder. It's going to accelerate over the course of the assay, so it gets harder and harder for the mice to stay on. Again, this is asking the mouse to perform a coordinated activity where different muscles are involved. And again, if you watch these mice, you can see the untreated and the AAV control treated, meaning it got a virus, but that virus did not contain a suppressor. Those mice are not really able to maintain speed with this rotating rod. And one of them has fallen off, the other one is about to fall off. But the wild type mouse on the left with the orange arrow and the suppressor tRNA treated mouse way over on the right-hand side, both of them are actually going to stay on this rotor rod toward the end of the assay, all the way to the end of the assay. And again, this is one example, but you can see the statistics in the graph on the left-hand side. In purple, we're getting very close to wild-type levels of performance with these mice, certainly a huge improvement over the untreated cohorts. So I think this is very exciting. It speaks to the importance and the power of restoring full-length dystrophin protein. We can, of course, look by various assays, Western blot immunohistochemistry, and see how much protein is in these mice. And we see protein is being restored in all the muscle groups that we look at. So if you look at the graphs on the left-hand side, we've got gastrocnemius, we've got quadriceps muscle. This is just two examples, but we see full-length dystrophin restoration in all the skeletal muscles we look at. We see it in cardiac muscle. We see it in diaphragm. And then on the right-hand side, you can look at various indicators. These are serum markers in this case of muscle injury. So we've got a myosin light chain and we've got a troponin. So both of these are extremely elevated in the untreated cohorts of mice, the light gray and the dark gray. You can see that both of them begin to drop down substantially with suppressor tRNA treatment. In addition, I don't think we've got the data to show here, but we've also looked much more broadly at the proteomes of these mice. And you can see pretty substantial dysregulation in the untreated mice. So lots, in addition to dystrophin not being there, that has downstream effects on many other proteins in the muscle cells. They're abnormal. Some are high, some are low. Some are proteins that interact with dystrophin, some are indicators of inflammation, muscle repair. And what we see on proteome-wide level is all of these dysregulated proteins start trending back towards wild-type levels. The ones that are elevated go back to normal levels. The ones that are low start trending upward. So we think the whole package really speaks to the profound impact that we're having in this mouse model by restoring dystrophin with suppressor tRNAs. So all the data that we've been looking at so far is for one specific class of suppressors. As Daniel and Elizabeth were alluding to, you've got three different types of stop codons that could constitute a nonsense codon: TAA, TAG, TGA. So the data that we've shown you so far is with a TAA suppressor, because that is the mutation that's in the standard MDX mouse model that everyone works with. But we're well aware of the fact that for bringing this to the broadest possible patient population, we need to address all the stop codons that are out there. So we are working on suppressors, and we've gotten very compelling data right now for ones targeting all three classes. And this is just showing you how the different types of stop codons, TAA, TGA, TAG, and green, and each one of them, of course, could be paired up with a different amino acid that's changed in that pie chart. So exact correction is what you'd get if we are putting back the exact same amino acid at the nonsense site that is supposed to be there. That's probably the most straightforward, and it's probably what we'll be going to the clinic with first. But dystrophin is one of those proteins that, now we'll show you a little bit of data for this, I think also allows for flexible correction. So if you've got a truncated dystrophin protein, even if that mutation is at a site that's got an arginine or a glutamine amino acid and it stops, we believe you're going to have a very profound and positive impact on patients as long as you're able to read through that, even if you're not putting back the exact amino acid that's supposed to go there. So this is what we call flexible correction. If we want to go to the next slide, just looking at exact correction, all the data that we're looking at for the TAA glin suppressor, we can now basically take our optimized suppressor for TAA, and we can just change that stop codon. And it looks like all the optimization work that we did for TAA carries through and gives us quite potent suppressors that are going to allow us to target in an exact manner glin TGA and glin TAG mutations. So that's already a very big expansion of what we'd be able to treat in an exact manner. But I think flexible correction is still something that needs to be considered longer term, because there are 19 different ways that coding codons can mutate to stop codons. And developing all 19 different products is a fairly long-term process. And I don't think it's necessary in cases of large structural proteins like dystrophin. So in a flexible correction, you could really probably address the entire nonsense patient population with just 3 suppressors. So let's take a look at a few more suppressor flavors that we've worked with in the mouse model. So here we've got an arginine TGA suppressor, or sorry, a glin TGA suppressor. And you can see that we're getting nice rescue of the protein with this. Actually, let me back up. The suppressor in this case is an arginine to TGA. It is doing the correction at a glutamine site that has had TGA mutation. So it is flexible in the sense that we're putting back a different amino acid, and yet we are still seeing very nice recovery of the full-length protein. And we've tried this with a few other flavors of suppressors as well in TAA mouse models. So this is, again, flexible in the sense that the amino acid of the site is a glutamine, but we're getting rescue with additional classes, serine and tyrosine suppressors. So we're seeing some very early indications that this flexible correction strategy works well in the mice, and it's something that we'll continue to follow up on. And here's some, basically it's an analysis of clinical data that's out there that supports the idea that for the dystrophin protein, flexible correction is probably a very powerful option. So if you just look at what the dystrophin protein is doing, it's undergoing a lot of folding and unfolding. It's a large structural protein that acts in some senses like a shock absorber in the cell. So all of these folding and unfolding domains are ones that we feel like are going to be very tolerant of flexible correction because they're already folding and unfolding in the normal processes that the muscle is going through. As long as you don't truncate the protein at those sites and get read through, you're almost certainly going to get a dystrophin molecule that does what it needs to do at the functional level. But you can also look at the actual mutations that are reported clinically and see whether they are pathogenic or non-pathogenic. So if you look at missense variants in DMD, they're very rarely considered pathogenic. So this tells us that having a missense mutation at this site really hasn't impacted the function of the protein significantly. And that again speaks to the fact that if you were able to do a flexible correction strategy with the suppressor tRNA, you'd be very likely to recover full-length protein that's doing what it needs to do. And you can look at this data in different ways from the clinic, but all of them sort of point to the same thing, that the dystrophin protein is tolerant of these types of changes. And next slide. So just to sort of give a summary of all the data that we've walked through so far, we're advancing suppressor tRNAs toward the clinic with the objective of treating 100% of patients with nonsense mutations. In mouse models of DMD, we're seeing restoration of full-length protein that leads to very dramatic restoration in motor function. It's blocking muscle injury, and it's doing all of this by allowing the cells to make full-length dystrophin following systemic delivery by AAV. We've already got in vivo proof of concept for a number of different classes of suppressors that would enable exact correction for a very large percentage of patient population. And we've got a lot of evidence indicating that flexible correction is actually going to allow us to expand even beyond that without necessarily developing 19 different classes of suppressor tRNAs. And as Daniel alluded to, we are very patient focused. So we're working very hard to create a robust safety package that will enable clinical entry. And we're starting to talk to the FDA about our approach and what those clinical data packages would look like.
And just as a last thing, this is data that we really haven't shown anybody yet, so this is very exciting. We wanted to share it with the broader patient population. Essentially, what we've been doing and what we've shown you today is work that we have done with some of our suppressors, specifically suppressors that we have tested at various time points, and the data that we've shown you is a 12-week time point. So this is data with some of our new suppressors, and right now we're using systemic delivery of suppressor tRNAs, and we're looking at that exercise tolerance. So this is actually rotarod data. I don't know, Julian, do you want to go on camera and talk about your data? Julian is the project lead, and he's worked incredibly hard with his team to drive this program forward.
Sure. So in this study, this is an IV study that we started approximately 4 weeks ago now. In that study, we took all the suppressor we could identify through our IM screening, intramuscular delivery screening, and all the suppressor tRNA you can see here were showing 100% of full-length dystrophin rescue. So we took those guys into an IV study. And after 3 weeks, I did some motor function experiments. Here is, as Peter mentioned earlier, some rotarod assay where basically the mice go on a wheel and run with an accelerating speed. And here you can see that we have different groups. So we have the wild type groups and treated MDX group. We have a control virus, which is a scramble virus, control vector. and you have like 5 different suppressor tRNA. And for each one of them, we can see some significant improvement after 3 weeks post treatment. So it's very, very encouraging for us. We're going to take down this study in about 2 months from now, and hopefully everything you saw earlier with our generation 3 suppressor tRNA will be much increased with this panel suppressor tRNA that we are currently testing.
Thanks, Julian. We're really excited about this data, and I think excited across all the data that's been generated. We're really open to any questions from the patient population and broader community.
Elizabeth, thank you for that. Peter, Julian, and Daniel. It's really impressive to see how that there's matured through each of the different iterations, and to get up to 60, 70, or even 100% dystrophin levels is almost unthought of. So congratulations on the data, and I really appreciate that. I want to invite on Leanna, who can join me for the Q&A session here. And if you don't mind, I'll kick off with the very first question that's always at the top of my mind. What's your timing to nominate a candidate, and when can we expect this to go into the development mode?
Yeah, we're as you know, we're advancing as soon as we can. So we aim to nominating a development candidate later this year, before the end of the year, and then advance to our IND-enabling studies and to the clinic.
Excellent. Thank you. Like, we're really excited. The data really speaks about just a uniquely different approach from other ones that we've got, going after full-length protein in individuals that really don't have options out there. So very excited, to see this here mature. Leanna, have you got some questions that have come on?
Yeah. Well, let's start with delivery. I feel like that was top of everybody's mind. So, I'm going to put some questions together here that come in. So since you're delivering with an AAV, individuals who have received Elevidys or any other AAV delivered gene therapy in a clinical trial would not be eligible. Likewise, right, if they received a dose of this, we don't have strategies yet to be able to redose this as well. But there's also some questions about what considerations are you thinking about? And this is also not just a Tevard problem, it's AAV in general, about individuals who may have neutralizing antibodies due to environmental exposure. Or are you looking into other delivery methods, for longer term?
Yeah, I would answer that yes, we are definitely looking at other delivery methods. So we have chosen AAV as our first approach, but all of our design process has actually been focused on being able to have a vector that we can drop into any AAV vector, but then also drop into a nanoparticle delivery or some other non-viral delivery approach. There's a lot of exciting approaches right now. And so we're keeping our eyes open, and we're open to collaboration with other companies so that we can get this to the patient as quickly as possible and allow as many patients as possible to access this type of therapy.
Yeah, Lena, let me take your question and sort of expand it a little bit, if I may. What about individuals who've been on, for example, one of the approved exon skipping therapies, and they've got a nonsense mutation? Would they be a candidate for this?
Yes, definitely. We could certainly have that approach after exon skipping, or you could have our approach and deliver by AAV and then take an exon skipping approach. I think that the only concern there would be monitoring for potential dystrophin immunogenicity. I don't think there's been any studies, and so we would have to look at that preclinically.
Yeah, we haven't seen that as much of an issue actually in the exon skipping world, but you're quite right. Can I also ask about, like, it's exciting to see the field here mature from the first generation results, the second generation, the third, and beyond. What generation would be the clinical candidate?
Generation 4 right now. So that is actually the data that Julian just showed you. And in particular, we are looking at a variety of different capsid options So specifically looking at AAV that are focusing on heart, skeletal muscle, diaphragm, and then liver avoidance, really looking at that right now. We actually have an ongoing study in non-human primates right now, and that will be focused on choosing a capsid that we could potentially move forward with.
Excellent. I got to say that it was, for me, really exciting to see the wild type like functional activity that came out of this only after 12 weeks of treatment. That's quite remarkable. I hadn't seen a lot of data like that presented before with other modalities.
Yeah, we're really excited about that, especially because we are able to go down to a low dose of AAV and still see this type of efficacy.
Yep. Any other
I'm glad
questions?
that you mentioned the non-human primate data because the question did come in on whether you're going just from mice to patients or you're planning to do a larger animal model. So that work, the fact that that work is already in progress is wonderful. Switching gears a little bit, there's some questions here about, whether the treatment as currently designed would have any effect on dystrophin in the central nervous system, because as you know, some individuals with Duchenne have CNS aspects of manifestations of their disease, cognitive difficulties or ADHD. And so this is of something which we're not currently addressing very well, I don't think, with a lot of therapies. And so maybe you could speak to that a little bit.
Yeah, it's a great question. We just recently, as a team, attended the American Society of Gene and Cell Therapy, and we were interested to see how people are approaching this from looking at the dystrophin isoforms that are in the brain. That is not the dystrophin isoform that we would particularly be looking at. We have looked at potential nonsense mutations in this form. We're currently not targeting the brain with our AAV approach. We have looked specifically at capsids that would be looking to target skeletal muscle, cardiac muscle, and the different types of muscles, but not necessarily crossing the blood-brain barrier. That could be something that we would approach with a nanoparticle delivery approach or some other mechanism. It's not a current focus, but it is something that we are watching closely, and we're aware that it's a need of this patient population.
Very good. Can I expand upon that, Leanna, if I may? One of the things that I would find attractive in there, Elizabeth, would be to consider muscle satellite stem cells, and seeing correction in there. And that could be like a particularly unique focus of some of the questions you might ask.
So we are looking at that. So that is an active topic of discussion at the company. Specifically, are the capsids that we are looking at able to transduce or infect muscle satellite cells, and how can we measure that functionally?
Excellent.
So it's something that we are really interested in. I was excited to see that some of the data coming out of the ASGCT conference showed that AAVs can get into muscle satellite cells, and it was very exciting to us.
Yeah. And that may have its own significant benefits on its own with muscle regeneration.
Yes. Yeah, we believe that that is the case, especially for a long-term benefit. And it looks like the satellite cells are critical for actually reduction of inflammation, as well as this exercise tolerance piece.
Yeah, I totally agree. And I feel there's also an opportunity to ask questions about disease regression. At that point, whenever you've got something with a full-length protein plus satellite cell activation again.
Agreed.
Yeah, that sort of speaks to some other questions that have come in, and I know it's still early days, but would you expect to see better effects in younger mice treated, translating to younger individuals? And have you started thinking about what the clinical trial might look like eventually?
We have definitely started talking about the clinical trial. We're actually working with regulators right now to actually, we're preparing our Interact package right now. This is a new technology, and it's guidance of the FDA as well as the EMA to go into these early meetings where you can ask questions about the technology and really lay out the regulatory plan so that things go as quickly as possible. I think that our goal is to get some answers and then move forward. We have talked about going in as early as 2 years old. And that is an option potentially, especially with the introduction of newborn screening,
Mhm.
so that we would have the advantage of newborn screening in many states right now that would identify the patient population earlier. We would have to establish safety probably in a slightly older population first. So 4 years to 12 years is what we're thinking right now, and have that be an ambulatory population because of the potential safety concerns that have arisen. I see questions about the AAV. It is not the same AAV that was used with Elevidys.
Yeah. And when you're talking about safety concerns, you're not talking about Tevard's data, you're talking about
Correct.
other
We're talking about AAVs in particular and the sensitivities that the FDA has around that.
Yes, in some older and non-ambulatory population that
Correct.
we've seen
Yeah.
in the field. Yep. Just wanted to clarify for you
No,
guys
100%.
as well as
Yeah.
everyone.
No, 100%. And we're very aware that just new therapies in general, there can be safety concerns. And so we want to attack it from all angles and make sure that we're not leaving any stone unturned.
That's great. I have one more very forward-thinking question. So this is like big long-term for everybody listening. Is this approach something that could end up working for, you could also adapt in some way to work for like single addition or single missing amino acids as well? Is that something that it could address other types of very small mutations?
Going on.
Yeah, this is a question that we get a lot. So our current approach really focuses on nonsense mutations. There are other therapies out there that could potentially do this individual correction. And this is not that approach right now.
Thanks.
Good question.
Great question.
Anything else, Mike, you've seen?
No, I'm just going to wrap it up.
Excellent.
So I want to thank everybody for joining us for the webinar today. And a big thank you to Daniel and the team at Tevard for the important work they're doing targeting nonsense mutations. It occurred to me as we're talking through the Q&A, there's literally thousands of human diseases caused by nonsense mutations. And I personally am super appreciative of the team that Duchenne is a major focus for Tevard. This is an important approach for Duchenne, and there's a clear unmet need in here. We look forward, obviously, to the future updates from the company as they continue to advance and work with the FDA. A post-webinar email will be sent out to all attendees tomorrow, and we'll let you know when the webinar is online. So from that, I want to thank everybody for joining and have a great rest of your day. Thank you, guys.
Thank you,
Thank you very much. Have a great day.